like to introduce Dr. Lonnie Russell. He is a professor at the Department of Physiology at Southern Illinois University School of Medicine at Carbondale. And many of you might know him via the book on the assessment, the histologic and the microscopic assessment of the testes that he has written with Dr. Atlin, who is a Sandoz person, a former Roche.
And thank you for coming. I'd like to do a little survey before we start of a few things. How about of those people in the back row? Are you able to hear me well? Okay, so thumbs went up, some fingers, I don't know which. But the second thing, how many of you are regulated by EPA? I'd like to know that. A very small minority, 2 people in the group. The rest of you are FDA regulated, I would suppose. Okay, so. We do have a couple of private practices. Okay. People in academia as well. Okay. I was sort of told that I should be talking from back here, and that doesn't seem to be too good. And I hope that, is it all right with the filming and everything that right here is a good spot? Okay, good. Okay, with that, I'd like to begin. I'm scheduled to be talking to you for about 2.5 hours this morning with a break. I know that Dr. Clegg has about 1.5 hours this afternoon. Dr. Clegg will tell you that he's not representing the views necessarily of the government. I'm representing my own views that have come about initially because of basic science research, and then becoming toxicology and pathology sensitive as we've done training courses here and in Europe. What I would like to do in the next 2.5 hours is to talk about an age-old type of testing, which is the mainstay of tox testing, is the histopathology, safety assessment, and see how we have now re-looked at it. I know many of you are re-looking at various systems in the body. You are told by people who are nervous system people that you have to become specialized in nervous system studies. You're being told by blood people that you have to become specialized in hematology. And I'm really no different. To understand the testis in many ways, you have to some degree become specialized in testis histopathology. And how you do that is largely dependent upon you and your company. So what we'll be talking about today is an old topic that we're going to revisit with new techniques, new ideas, and new approaches. Now, why is histopathology of the testis so important? The word "of the testis" should be in there. And I've had 4 major rationales for thinking that this is a special area for you to learn and to become, have an increased awareness of. There is an increased interest in reproductive toxic effects in general and in male reproductive effects specifically. If I were to ask this same question 20 years ago for pathologists here, hey, we just put on the slide, as long as the testis is not wiped out, that's okay, we just go forward. Male fertility is not necessarily important. Well, it has become important. We realize now that approximately 50% of causes of infertility in the male or infertility are due to the male. And so there has been an increased awareness of male problems. Testis histopathology may lead to provide valuable information that will lead to valuable mechanistic interpretations. That's not something that's generally normal in pathological world to try to determine mechanisms. But what I will try to show you, especially later on this morning, is that you can have some idea of how mechanisms, what the compound that you're using has an effect, what it's doing, and how it's working by looking at testis histopathology. Testis histopathology is a means to localize reproductive effects to a problem with sperm production. It is especially important nowadays that there is a trend for shorter-term testing protocols, both in the FDA and the EPA. Testis histopathology is currently inadequate from the standpoint of the training given to the histopathologist. I guess that's why we're here today. And the methods used to assess testis histopathology, in particular the tissue preparation protocol for testis. So, although we have people who are regulated from different agencies here, the endpoints that are generally in current use are very insensitive endpoints. Breeding studies. They're very insensitive for risk assessment in that a rodent model, for example, you can deprive or knock out spermatogenesis somewhere in the neighborhood of 85% off normal and have only 15% active spermatogenesis and still have 100% fertility. So this is a very insensitive measure of toxic effects. Testis weights. I like testis weights. They're a very good general predictor of testis function. But again, they're insensitive. I'll show you some data of people, of how many animals and so forth that you have to have later on this morning and what the power of the test is. And testis and epididymal histology, the mainstay of the evaluation of the testis. But current tissue preparation methodologies and the level of training of the histopathologists make it an insensitive measure of testis function. Let's take a look at our guidelines. Guidelines that in some cases may be changing with time, especially the FDA guidelines. And in some cases are only draft guidelines and not finally approved. But as you know, even draft guidelines carry a lot of weight. Here, for example, we pulled out a portion of the EPA guidelines. The quality of the information presented from histological analysis of spermatogenesis improved by proper fixation and embedding in tissues, including the use of staging. I've outlined that, and I haven't told you what the previous sentence says, but that word rings a lot of bells. Let me ask Eric in the back of the room. Eric, is this the current wording of the guideline nowadays, or has this changed somewhat since then? Okay. FDA guidelines. The agency encourages the use of good pathological histopathological examination techniques in the repeated dose toxicity studies in addition to the staging of spermatogenesis, which is routinely employed. Very scary statement to me that was written by FDA. One that is poorly worded, was written by an individual who does carcinogenesis work, and which can be interpreted in varying ways, and which I would like to give my interpretation for you a little bit later of what this means and what it means to get the right answer from your tissues, or the best answer from your tissues. So these are some of the guidelines that we have to work by. It is impossible for me to talk to you about new techniques in evaluation of the testis without going back for us to understand spermatogenesis. And if you will remember, many of your veterinary pathologists, and I would suppose some of your toxicologists, how many toxicologists do we have in the group? Okay, just a small number of toxicologists, and I would assume most are veterinary pathologists. But when in veterinary pathology school, when you were trained about the testis and understanding the testis, generally the instructor could not readily point out cell types to you. Probably they did not identify the Sertoli cell correctly. They probably could not tell you what were Spermatogonia and when they transitioned into spermatocytes. It's because there has been a general lack of knowledge of spermatogenesis out there in the field. And that is perpetuated in veterinary training in veterinary schools and so forth. I think with time this is now getting better and better and better. But what I would like to do is tell you what some of the things that mainly are in the book that was written by myself, Dr. Etland, my postdoc, Dr. Sinha McKeown, and Dr. Clegg, who will be your speaker this afternoon. For the toxicologist in the group, we're going to start right here with this organ down here. And we will proceed to actually we went from bull here to actually going to rat testis in this situation here, an unfixed rat testis and a fixed rat testis. in which if you section here, and this is a dog slide, you will see that there are tubules in the testis. The epididymis here is attached. It is the reservoir for sperm, as well as the region which motility is acquired and fertilizing capability. We're going to be focusing primarily on the testis. In a rat testis, we see that we've isolated one tubule. It is a loop of tubules that joins at either end to the rete. The rete being the initial collecting duct that will proceed, ducts from the rete, afferent ductules will proceed out of the testis to the epididymis. But this loop here makes bends in this manner, which are shaped overall in a rat much like a cone. Put a little ice cream in there if you wish, and this gives rise to the thought, should we section in histopathology the testis longitudinally, or should we cross-section the testis? Many people have specifically targeted one or the other way. The testis should be cross-sectioned so that you see round tubular profiles. And that doesn't mean it should be cut open right at necropsy. It just means at the time of sectioning the tissue, you should perform cross-sections. That'll become apparent to you a little bit later on. These seminiferous tubules here in the scanning electron micrograph are separated from the intertubular cells here by a space in the rat. This is the lymphatic space. This area is the lymphatic space. There are vessels which run in the intertubular area. The little round cells here are Leydig cells, but most of this looks relatively smooth. Since this is a lymphatic space in here, we have an endothelial covering that covers and hides most of the lymphatics in this particular slide. Okay, getting higher and higher resolution in this tissue, which is a methacrylate embedded tissue. We see that the great majority of the volume of the testis is occupied by the seminiferous tubules, a small portion by the Leydig cells here, and this very dark space here is the lymphatic space that is present. The lymphatic space carries proteins. If you were to take a 50-gauge needle and inject it something directly into the testis, for example, a dye, within seconds the whole testis would turn blue because of the lymphatic space allows the spread of the material throughout the whole testis. So here's this complex procedure that many people are afraid of, spermatogenesis. And I will ask you to bear with me for the next hour or so in describing spermatogenesis and going on from there. It's not so tough as people realize. You just have to break it down into components and review it with time. There are three cellular phases of spermatogenesis. So I break the entire processes down into three phases: spermatogonia, spermatocytes, and spermatids. Spermatogonia, think of functionally as mitotic divisions. Think of that because each rat, for example, each testis of a rat will produce somewhere in the neighborhood of 20 million sperm per day. Okay, so you need lots of mitotic divisions. And this is where it occurs, in the spermatogonial population of cells. Spermatocytes. Meiosis ensures genetic diversity. Okay, in terms of meiosis, we have the process of recombination or crossing over taking place, so that the chromosome that you got from mom and the chromosome that you got from dad exchange material to produce chromosomes that are absolutely unique, so that the gamete has a unique chromosome that has parts of both the male and female from the past. Now, this little diagram describes the three phases that I talked about: spermatogonia, spermatocytes, and spermatids. And if we start off here, each of these arrows represents a cell division. And so there are cell divisions that are very common among the spermatogonial population of cells. And when we get here, we have fewer cell divisions from this point on. This is the spermatocyte or meiotic population of cells where the chromosomes are doing something in the crossing over process. But we do have cell divisions that occur at the end of meiosis, two very rapid cell divisions that occur to produce spermatids. There are no cell divisions after the spermatids, but there is a very amazing transformation that takes place. A round, very round, typical appearing cell goes to one that looks like something like this in the rat. It's a cell biologist's dream because many, many things are happening. We can look at them in four ways. One, there is a tail forming, and that's going to ensure motility. The second one is there is an acrosome forming. That acrosome is this dark area here, which we will speak about with time, and that acrosome ensures the ability to penetrate the egg. Thirdly, we've got a bulky cell here. Here we have one that can move and swim. We have to condense the nucleus. Now this is not an accurate representation in that the actual head is much larger than it should be here. But this sperm cell here is only a fraction of the size of this spermatid up here. So the condensation of the nuclear chromatin, the histone changes that take place, and the shaping that takes place is very important. The fourth process is elimination of cytoplasm. Cytoplasm comes off in a blob known as the residual body. The residual body. Residual bodies have often been mistaken for degenerating cells by pathologists in the past. Let's start over again with the spermatogonial population of cells and go through this again because we will have some significant things we want to talk about that relate to pathology. Here are two spermatogonia. The spermatogonia are connected by intercellular bridges. As soon as the stem cell divides, it will become committed to spermatogenesis. It has a unique process that occurs in which cytokinesis is not complete. Incomplete cytokinesis leads to the formation of the intercellular bridge. Now, people have asked, why have this process in the testis? The process is there because these cells will now develop synchronously because any messenger RNA, any transcription messenger RNA is going to be able to go between cells of a clone. And we have a synchronous development of cells such that all sperm are matured exactly at the same time because of the sharing of cytoplasm. Now, what is the kinetics we're talking about here? We have lots of spermatogonial types. I don't want to bore you with that. But if we begin with the observed linkage of 16 spermatogonia together, not necessarily in a linear chain as shown here, but linked nevertheless, we can look at other types of spermatogonia, and here are the numbers that are linked together theoretically by intercellular bridges, such that by the time you get to spermatids, which are not going to divide anymore, that's the potential number of cells in a clone. You ask yourself, when I kill cells due to a toxic effect, do I kill a whole clone? And the answer is generally no. Rarely our clones go completely. Usually what will happen is that they will pinch off their bridges, one will die and one will not. If you have a very severe toxic effect which would have killed the cells anyway, you'll probably wipe out everything. But in many of the types of things I'll be talking about today, which are not wipeouts of the testis, you kill only parts of a clone. Meiosis is probably the toughest thing as you came across in veterinary school when you studied spermatogenesis. I think I can try to make it as simple as possible. The names that are used for meiosis are describing what's happening to the chromosomes during this process of recombination or crossing over. Preleptotene is a significant phase because now we're in spermatocytes. We've forgotten about spermatogonia. We've moved on to spermatocytes. So this is the youngest spermatocyte here, and this is the DNA synthetic phase, where S phase occurs, and you essentially replicate your chromosome. This, in fact, is the last S phase of spermatogenesis. That becomes significant in understanding toxic effects in some cases, knowing that there is not another S phase occurring. It occurs very early in cells that are right at the base of the tubule, and then there is no further S phase. Leptotene. This is the first step of prophase. The chromosomes appear thread-like. Homologous chromosomes are not paired at this point. They are not paired. Zygotene is the first pairing of homologous chromosomes, such that they appear thicker at this point. And at pachytene is when the actual chromosomes cross over. Chromosomes actually appear even thicker at this point. If you take homologous chromosomes and you have segment AB and AB, you can actually exchange such that now small ab, which was over here on the green before, we should have this green and this part yellow right here, there is exchange of this segment, which is crossing over or recombination that occurs. How long is pachytene? Pachytene lasts an awful long time. For the rat, it approximately lasts almost 13 days. It's a very long process. Preleptotene, leptotene, zygotene together are not even half the length of this process of pachytene. Pachytene cells are in almost every tubule that you will find. The steps that follow are very short. Diplotene, we now lose the chromosomes that were paired together. They come apart, and one of the two chromosomes will go to each daughter cell in meiosis 1. In the secondary spermatocytes, they're almost not around at all. In the rat, they only exist if you talk about a 53-day cycle in the rat, or not cycle, 53-day length of spermatogenesis in the rat. Secondary spermatocytes are around only 9 hours of this period. What does that mean? They're hard to find. You almost never see them in the seminiferous tubules. And if your instructor in veterinary school said, here's primary spermatocytes, secondary spermatocytes, and spermatids, and told you that for nearly every tubule, that instructor was wrong, because they're very rarely seen. They're very short-lived. And meiosis 2, the sister chromatids separate. The final result being that you have haploid cells that are now called spermatids. So we're moving on to the third phase of spermatogenesis called spermiogenesis, where the spermatids are present. I just want to check and make sure that my voice is carrying to the back of the room. Is it okay in the back? Okay, thank you. Sorry. This drawing by Dr. Claremont shows the evolution of a spermatid. There are no cell divisions, as we said earlier, and this cell, which is typical of many in the body, essentially becomes this cell, which is a very highly specialized cell, and goes through the processes that we talked about a minute ago. And in particular, it develops an acrosome. And I want to give you some terminology in relationship to the acrosome that will carry us a little bit further into the talk. We're only looking at the nucleus here, and our Golgi apparatus produces a proacrosomal vesicle, like a secretory granule, and a proacrosomal granule, which is the material inside the vesicle. These coalesce, they bind to the nucleus and spread out over the nucleus to become the acrosome. So the terminology we will often talk about is acrosomal granule, acrosomal vesicle, and acrosome. This process of acrosomal development and acrosomal spread is one in which is continuous, but allows us to separate different phases of the time the acrosome is spreading over the nucleus, and to categorize this whole process, and to come up with what we call stages that I will talk to you about in a minute. So it is an extremely important process. Here is an actual electron micrograph. I don't think we need it darker, but should it be darker in this room, it would have shown up a little better. The acrosomal granule and the acrosomal vesicle, the nucleus, and the Golgi apparatus. When you are looking at a slide, don't confuse the Golgi with the acrosome. We'll show you more on this later. Ah, but you say to me, I can't see the Golgi normally in my tissues. Well, that may be true. Maybe we don't have the right fixation techniques or staining techniques to show these structures up. We're still talking about spermatids. We haven't talked about the Sertoli cell yet. We will, but I will simply say that the spermatid at one point in development, here's its acrosome here, Golgi, is randomly oriented. That acrosome can point to anywhere. But at some point, it orients toward the base of the tubule. The acrosome points downward. It makes sense because all of the tails are going to be pointing into the lumen of the tubule, and we're going to get a coordination of spermatogenesis to where all of the cells are packed, heads down, tails into the lumen, instead of a random orientation of things which would be impossible to coordinate with time. Now, I've talked about flagellar development, and flagella can be very complex structure. I don't want to go too much into flagellar development, except to say that the flagellum is complex with different parts in a very complex process that will form the flagellum. The flagellum forms in a very young spermatid from the centrioles. And an outgrowth of the centrioles, which would be an axoneme. For those of you, it's a 9+2 microtubule arrangement. And at some point, those centrioles impact on the nucleus. And after that, we get the accessory structures developing. Here, mitochondria surrounding the flagellum that will form the energy source for flagellar movement. Head shaping. Each species has a characteristic head shape. One of the most common pathologies is defects in head shape of spermatids. This happens to be, by gosh, I don't know. It's either a rat or a mouse. I believe it's a rat. They're very similar at this phase of development. But the acrosome is here over the head, and the tail is beginning to come out at this point, and this structure is called the manchette. It is a microtubule-based structure, and we can influence head shape in some cases with compounds that affect microtubules. Our lab has shown to a large extent that the manchette does govern, is responsible for head shape in the normal animal. If this is messed up, you have defects in head shape. Chromatin condensation begins both in the mouse and the rat, in fact, all species that I know, from the anterior aspect, the tip, and proceeds caudally. You notice this is much more dense than normally, and we're actually beginning to get chromatin condensation taking place here, as well as elongation of the nucleus. Okay, chromatin is fully condensed in this spermatid. Oops. At this point, get my button straight. And we're about release. We're about ready for release of the spermatid. At this point, the spermatid develops a number of strange and unusual structures that are probably involved in sperm release. I've called them the tubulo-bulbar complexes. This is the Sertoli cell surrounding the spermatid here, and again the Sertoli cell here, and here are the tubulo-bulbar complexes. I point this out because sperm release is one of the most critical sites where toxic effects can take place. And I will be asking you to pay special attention to this as we go along. After sperm are released, we have the structure left that is a residual body that can look much like degenerating cells. If this kind of a picture is not what you're familiar with, I'm sorry. I do a lot of research, histopathology, and just basic research into normal spermatogenesis. This is an epoxy section at 0.5 micron where we can see mitochondria, we can see the Golgi apparatus, and we can see even details within the residual body. The role, we haven't discussed the somatic cell within the seminiferous tubule yet, and that is the Sertoli cell. The Sertoli cell, I'll make the statement now and probably make it several more times as time goes by. This is my opinion, and I think it's conservative. Over 50% of the effects that one sees in the testis are probably Sertoli cell-based effects. Over 50% of the effects you see. Now, that's not something that you're probably used to thinking. You're thinking about germ cells being affected. Because after all, if you wipe out the testis, you still have what left? Sertoli cells left. Those must be good guys. You know, you can't hurt them. Well, that's not true. What seems to be the case is, yes, you hurt the Sertoli cells. They are impossible to kill, but they're hurt enough so they cannot support the germ cell population. The germ cell population is wiped out, and the Sertoli cells are staying there saying, hey, I'm smart after all. I can survive. And as I said, 50% of the lesions, I believe, are due to Sertoli cell defects. If you see vacuoles in your epithelium, large vacuoles, that's a clue to Sertoli cell defect. If you see failure of sperm release, that's a clue to a Sertoli cell defect. And there are other clues that one may have, such as nonspecific degeneration of germ cells. One here in one stage, one over here in another stage, just a very unhappy, disorganized epithelium. That's a clue to a Sertoli cell defect. Have we proven this? I don't think there is extremely sound literature out there that proves all of these things are due to the Sertoli cell. But inductive reasoning indicates in many cases that it is due to that because of its role in organizing the epithelium. If you give a germ cell toxicant such as methoxyacetic acid, you kill Pachytene spermatocytes at a particular stage of the cycle, they all go real quickly. You know that's a germ cell toxicant. But when things go sporadically here and there, it is hard to believe that your toxicant can affect 50 different million places in the testis. It is more that the general nutrition provided by the Sertoli cell is interrupted rather than your toxicant having multiple effects. And that's the kind of reasoning I've talked about that leads me to my conclusion. Okay, the Sertoli cell. Recently edited a book on this topic, 800-some-page book, which no one had ever dreamed we had that much information on the Sertoli cell. But I would say in the last 10 years or so, there has been a voluminous amount of information on the role of the Sertoli cell. It is the key mediator in spermatogenesis. If you look at a drawing of a seminiferous tubule, here, this drawing done by my sister, and I have to say that every time or fall into disfavor, the Sertoli cell is this cell right here. You will not recognize it in your epithelia because we have exaggerated its width here. It occupies in the rat something like 16% of the epithelium. But it is throughout the epithelium, so most of its processes are very thin. It really forms the architecture of the epithelium in that it binds to a basal lamina here and binds to adjacent Sertoli cells. This region of binding here is very complex and is a tight junction region which forms what I call the Sertoli cell barrier. Other people have called it the blood-testis barrier, and I believe it's the only real barrier of molecules that are from the blood outside the tubule going to the center of the tubule. The Sertoli cell barrier is one of the tightest epithelia in the body, containing over 100 lines of tight junctions. And here we have Spermatogonia, which reside on the blood side of the barrier, and the remainder of the germ cells reside on what's called the adluminal side of the barrier. It has implications in your work because many of the compounds you give will affect Spermatogonia, but may not be able to reach the adluminal germ cells. A lot of that depends on the lipophilia of the compound, the molecular weight of the compound, and there are papers published by Lee and Dixon and so forth, that indicate which materials get through and which do not get through. So the Sertoli cell seems to have a very intimate relationship to every germ cell type in the epithelium, the spermatocytes, the spermatids, but especially the elongate spermatids, where the elongate spermatids are down inside the epithelium in crypts within the epithelium. And this occurs, as I showed you earlier, when the head orients itself within the epithelium. In the rat, this distance can be a half micron from the basal lamina. They are not within the cell, they were in crypts. And they will move near sperm release toward the lumen. This is a Sertoli cell we built in our laboratory, the first one that's ever been viewed externally. But you can see the spots for the germ cells, and you can see where the elongate cells come out the top. In the rat, the Sertoli cell has approximately 10 elongate spermatids that are associated with it. Each species has a characteristic number. Some are very high, some are very low. Human is about 2. There are some monkeys as high as 26. Rabbit is very high. It's a species-specific phenomenon. The Sertoli cell nucleus is about all you will see in light microscope sections. And you will see it because of its characteristic nucleolus. It's always on the base or slightly up from the base, but it is the cell that has little heterochromatin and a very characteristic, what we call 3-part or tripart nucleus. This is really DNA here and here. This is RNA, the nucleolus portion of it. But that's one way that you can recognize Sertoli cells. As indicated before, the Sertoli cells have 2 configurations, essentially. One where the spermatids are deep within the epithelium, and this occurs most of the time, and one where the spermatids are near the lumen, ready to be released. And that has numerous junctions of very unusual types that bind germ cells and actually help move germ cells within the epithelium. What is the hormonal control of spermatogenesis? It is a pituitary control, as most of you already know, in that the pituitary secretes 2 glycoprotein hormones. FSH binds directly to the Sertoli cell. It binds to the plasma membrane, elicits adenyl cyclase, and there we go. LH binds to the Leydig cells, elicits works the same way through an adenyl cyclase system to produce testosterone. The receptors for testosterone are where? Which cell type? The Sertoli cell. Probably none in the germ cell. So both hormones are binding to the Sertoli cell. And if you take away the pituitary, you will lose the vast, vast majority of spermatogenesis. So the Sertoli cell is the key mediator of the hormone response. Just like to take a little bit of a mental break for a minute because we're going fast, and ask if there's any questions about the first spermatogenesis. Either you're confused or I am perfect. Any questions? Well, then let's proceed on and talk about staging to understand spermatogenesis. For many of you, one tubule in the testis is just like another. It's not easy to tell them apart because you haven't taken the time to sort out which cell type is which cell type and which proceeds to the next, and made some ordering or made sense out of it. This was done actually at the turn of the century, and more recently in 1952, categorizing stages. And even more recently in the book that I mentioned to you that Dr. Clegg and I put out, that allows you to understand staging for spermatogenesis. And what I would like to do is to turn you on to the concept, but not teach you staging at this point. A lot of our understanding of spermatogenesis is by understanding how the Golgi apparatus elaborates the acrosome and what the acrosome does with time. And here is the acrosome, binding to the nucleus here with a proacrosomal granule, forming one, and it spreads out over the nucleus. Now, so I will say right away that the primary determiner, could we have the lights down just a little bit for this current slide? The primary determiner of classification of spermatogenesis is by using the acrosomal system of the spermatid generation of cells. Question, can you stage if you don't use the acrosome? Yes, but it's a little more difficult. You can use other cell types if you're real good at it. In other words, when you have a depleted epithelium. But let's take a typical tubule, and I've picked one again which is epoxy embedded because we can see things. You will not be able to see a lot of these features on a paraffin H&E or even a paraffin H&E Bouin stain section. But let's take a tubule from a mouse in which we see cells throughout the epithelium. The first thing that was done to develop the staging criteria was to ask, what are the different cell types within this epithelium? I'm going to ignore the Sertoli cell with its nucleolus here and here because it's a somatic cell. We're going to be dealing with the germ cells. So we picked out the various cells that are in here. And starting from the base, we picked out Spermatogonia. And there is a Spermatogonium, the same cell here. We've isolated another type of Spermatogonia here, which is this cell type right here. Now, you'll see several of those. Spermatocytes, you will see several spermatocytes here. This spermatocyte looks like it is this one right here. This round spermatid, uh-oh, I'm in trouble. Somebody will have to help me find it. How about up here? There it is. And there are lots of these within the epithelium. And then the elongate spermatid is this one here. These are the cells that are at the same developmental phase within this tubule. So I can make what's known as a cell association. It turns out that every time I see a round spermatid with an acrosome developed as this one is, I will see the rest of these cells all coordinated in their development with that cell. That's called a cell association. All I have to do now is find out how many different cell associations are there and put them in the right order. And here's the basis, the rationale for cell associations. Here's why you have them. Now let's just start in the middle, the beginning of the circle with Spermatogonia, because I'm going to go Spermatogonia, spermatocytes, spermatids as I go outward from here. So we start with our most primitive Spermatogonia, and we go around this circle in time until we come to another cell, which is actually another Spermatogonia at this point. Well, what happens here as this cell continues its development around is that Spermatogonia were kicking in development every time. There are new ones kicking in. So that when this developmental stage is released, we get this kicked in again. When this developmental stage is released, we get this kicked in again. One of my colleagues, Larry Johnson, compares this to college. As soon as you reach your end of your freshman year, there are new freshmen coming in. As soon as you reach the end of your sophomore year, there are new freshmen coming in. As soon as you reach the end of your junior year, there are new freshmen coming in. And as soon as you reach the end of your junior year or senior year, there are new freshmen coming in. So the recruitment of cells that continues to occur with time helps build this cell association I talked about. And these lines that you see going outward like this, and the group of cells can be classified. A minute ago, we looked at stage 6. That's where we're looking at an acrosome about this well developed. And that cell association is present in a cross-section seminiferous tubule. Cross-section seminiferous tubule, and that's called a stage with a Roman numeral. we can construct a stage map, all based upon acrosomal development. Now, the fine details of the acrosome are not especially clear in here, but as we see, the acrosome is continuing to develop. Stage 6, for example, would be this stage here in the mouse. Whoops, I'm sorry, right here. And as Spermatogonia continue along this process, the way you read this map is you go from left to right, and when you get here, you jump up to here. When you get here, you jump up to here. And when you get here, you jump up to here. The Roman numerals are stages. The development of the acrosome is in steps. It's in steps of development. We say a step 2 spermatid is in stage 2. That was for the mouse. Here's for the dog. Someone came along and said, hey, I only want to make 8 stages for the dog. And actually, the dog is very easy to stage. It has this giant acrosome and very distinct head shapes. And sperm release occurs in stage 5 here. The frequencies of the stages. All the stages are of not the same duration. Remember that requirement from the FDA that someone go do staging? The FDA wrote that, and it has been interpreted by many people that the first thing the pathologist does is head to the microscope and see how many stage 6s you can find, how many 10s, how many 11s, and so forth. A total exercise in futility the way it is written. This stage frequency is only done by very few people. It is difficult. It is laborious. What I will tell you later on is that you should be looking at pathology the way you normally do under your microscope, a few minor modifications, with the knowledge of staging, but not do something called staging. The knowledge of staging allows you to find, once you find a defect, it allows you to determine where that defect is, what cell types. It allows you to talk to other people, because if you say it's step 6, hey, that's already defined. Everybody knows that. Even regulatory agencies can understand your reference of what step 6 is. It's a common means of communication. So again, spermatogenesis is done with the knowledge of staging. Not only are stages in different frequencies, but their frequency is related to the time in which they are present. Stage 7 was a high frequency, and it takes a long time. And we can time spermatogenesis, something that becomes important in toxicology I'll tell you about a little later on. Each stage has a different period of time that it's around. So far I've dealt with what is in cross sections of seminiferous tubules, but what is in longitudinal sections? One stage is in one cross section, but in longitudinal sections you will see many stages. That does not imply that for stage 7 to become stage 8, the cells have to move there. It just means that this 7 will become 8 in that section of the tubule. But we've got an end closest to the rete and an end distal to the rete. And the basic plan of the stages is they descend from the rete. Now, this doesn't have many toxicological implications, but it is often a question that is raised, and I want to answer it. So if you're arbitrarily starting with 10, 9, 8, 7, 6 as you go away from the rete. So there is a descending order of stages. The timing of spermatogenesis is critical for designing tox experiments and risk assessment, and as epididymal transit, how long it takes for these things to get through spermatogenesis. After all, if you give a compound and you wait too long, the effect, if you have a period where you're not giving the compound and you wait and all the effect is passed through the epididymis, you've missed it. Or if you give a compound, you wait too short a period of time, you're going to have to look at very, very young germ cells to determine where that effect is. You have to be very careful about that. So you need to know the timing to some extent. In the book we wrote, we did a lot of different species, but if we go to rat, for example, One cycle takes 12 days. Now, what do I mean by a cycle? Spermatogenesis is 53 days. A cycle on the map that you saw was from the left side to the right side. There are 4 or 4.5 cycles, depending on who you talk to, in spermatogenesis, depending where you begin with. So you take this 12 to 13 days. It's actually, in a Sprague Dawley rat, is almost 13 days. So you multiply that times approximately 4.5, you get close to 53 days in the rat spermatogenesis takes. Mouse, we're talking about a much shorter cycle length than the rat. This is some degree repetition. This is a cycle map of the rat, and I just wanted to show you how the Spermatogonia were in the cycle map, how the spermatocytes were in the cycle map, and where the spermatids were. Here are the Spermatogonia in the cycle map to this point. Here are the spermatocytes at this point right here in yellow. All of these are pachytene here. All of these are pachytene. Preleptotene, leptotene, zygotene, pachytene. Pachytene almost goes the whole cycle. There is diplotene in secondary where you have 2 meioses here. Spermatids in tubules are almost 2 cycles, actually 1.5 cycles. Well, look at the acrosome growing here. Right away, I'm not going to teach you staging, but I'm going to show you some things that would help you. If you looked at a tubule and it had 2 generations of spermatids, you're dealing with the left side of this map. I happen to know that these spermatids are ready to be released, and they're right near the lumen, where all of these are deep within the epithelium. That separates this from that. And all of these head shapes and chromatin condensations, although not shown well here, are relatively easily distinguished. How long would it take you to learn spermatogenesis in a microscope setting? Spermatogenesis, and I mean staging. It would take you 4 hours to be able to identify stages reasonably well, and then repetition after that. So it is a very possible situation. It would require a little bit of didactic work before that. But this is about the length of time that it would take. So as we said, the acrosome is the criteria for the primary identification of the spermatids. Now let's analyze this epithelium as sort of a task for our own cells. This epithelium happens to be from a hamster, but it really doesn't matter. Okay, what cell type am I pointing to right here? Sertoli cell has that giant nucleolus and very little heterochromatin around. We see cells of a like type here almost always have these pachytene cells with these very large chromatin masses. But this is the key cell, the spermatid. It has the acrosome, and here we have a second generation of spermatids, here with the heads with the tails present. 2 generations of spermatids almost ready for release. And this species is going to be very close to a 7 or 8 stage. That's about as simple as it is. I happen to know this is actually a stage 7 because, well, I'm not going to go into it because I don't want to get into too much detail. But here's another generation of cells. If we know this generation, and if we stage this one as a step 7, we know that's a preleptotene spermatocyte right there. We don't have asynchrony. Rarely does a chemical or compound cause asynchrony. When you have a cell association, you can predict what these cell types are just based upon what this cell type is. Here is the cycle map for the rat. The rat has 14 stages. And in the latter part of the cycle, if you're dealing with only 1 generation of spermatids, the head shape and the chromatin condensation are very characteristic. In stage 14, you have meiosis occurring. That doesn't look like a meiotic cell. It's that short-lived secondary spermatocyte. But it's easy to tell 14 because meiosis is occurring. Just a few hints. Staging is not impossible. It can be done in a very short period of time. In our book, we do give vertical cell associations so that we categorize exactly what these cell associations are at particular stages of the cycle. All right, Dr. Russell, how does this apply to the human? Well, I've got some bad news. The human has spermatogenesis that appears to be organized in a very different way. It's not clear how, there's a big debate in the literature of how human spermatogenesis is organized. When you look at a cross-section tubule, you can pick out as many as 6 stages in a human seminiferous tubule. In a rat, mouse, guinea pig, hamster, rabbit, dog, most monkeys, only 1 stage per seminiferous tubule. Well, in the human, what's happening, some people think, is that, that there is a spiral of a particular stage within the tubule. And then another stage spirals in there, and yet another spirals in there. There's now information that this spiral theory may not be correct. It is a very debatable topic at the current time. Does that make staging invalid? No, it doesn't, because you're stuck with animal models. You have to know the model you're dealing with. What makes the human different than the rat, nobody knows. Why this occurs, nobody knows. Human spermatogenesis is very inefficient. And there's all kinds of rumors out now that, for example, in the last 50 years, human spermatogenesis decreased by, what, 50% or something of that nature? People will say that humans are on the verge of infertility. I mean, because if you lose a sperm count in a human, you don't have to lose much to become infertile compared to a rodent, for example. So all of these kinds of things play into effect, and regulatory agencies get very nervous when they do extrapolation from animal species to human, because we don't quite know what we're dealing with at this point. Dr. Hess, a colleague of mine, has developed a software program. If you're computer-oriented, and for example, you're looking at the dog, here's the staging map with the cells. He's got pictures. You double-click on these pictures. It will bring up a particular stage of the cycle and allow you to sit at your computer and refresh your memory on particular species. He's got 23 species in here, many of which you wouldn't be interested in. But in this case, you can actually click on cell types. It'll tell you what it is. There is some predictability with lesion predictability that you can use given certain situational circumstances. That tells me that we pretty much finished the introduction on spermatogenesis. Just to summarize and say that spermatogenesis is extremely organized. And that organization shouldn't scare you, but allow you to take advantage. You can take advantage of that organization to better understand spermatogenesis. If you look at a liver cell, one liver cell might be just like the next one. If you look at spermatogenesis and you have a defect, you might be able to tell exactly when that defect occurred, what cell type, and know what's going on in that cell type, and maybe make some predictive advantage. What I'll be talking about in my next lecture is pathology, and we'll take some of this knowledge and go on and talk about pathology from then on. How is our time?
We're doing very well.
Very well, okay. I didn't even look at the watch, but thank you. I had several over the break. Yes, sir.
Lonnie, did you say that stage 14 was going to end up at about the reach by the time it reached there? Is that correct?
Okay. You have a misconception in that 14 will take place in all parts of the tubule. In other words, you're not having a movement of stages down the tubule, but that whatever stage is near the reat, the next one distal from the reat is going to have a descending order. So there is no movement of stages within the tubule. It's just that that, for example, a stage 2 in one particular part of the tubule will eventually start change to a 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Then it will go back to 1 in that same part of the tubule. Okay. At some point, there will be a 14 near the REIT, but it's only for a very short time.
One of the headings you had was timing of spermatogenesis and epidermal transit. Could you comment more on transit of spermatogenesis?
Okay. First of all, it's species specific. The question was, could you comment more on the timing of epididymal transport? It is species specific, but it can be changed by, occasionally there are some things that can change epididymal transit time. In the rat, I'm not sure exactly what it is, but I believe it's about 10 days. Do you know, Eric, exactly what it is?
We use 7 to 10 days, and of course ejaculation frequency.
Will influence the time, especially in the cauda, I believe, but not in the caput and corpus of the epididymis. Yes. Hi, how are you doing?
Lonnie, I don't mean to steal your thunder. I've heard your presentation, and it's wonderful. And I encourage all the pathologists who haven't heard you and Dr. Hess make your presentation to do so. It's extremely informative. But I have a question in going back and trying to apply this in drug safety evaluation in our department, where you have described changes in the stages for a particular stage, and then you can follow that in the maturation of that sequence in the testes. Is it not reasonable where you have an animal being continuously exposed to a drug for 28 days, which in the rat would be 2 complete cycles? Okay. Would it not be reasonable to classify the tubules in the testes as either abnormal stages or normal stages, and not really be concerned? Is there something? Am I thinking correctly or not?
Well, I think you're thinking of a good question, and I think I have a good answer to that. And the question came up in another way during the break from someone else. You know, why don't we just say atrophy and get on with it, you know, or degeneration and get on with it, because that's what we've always done. One of the answers is that it may not be acceptable in the future by the regulatory groups, okay? Another answer is that you are liable as a company for the product that you put out to be safe. And if you can provide information that assures your own selves that what you're seeing is something that is not going to be a severe damaging agent, because you can now describe it in depth, and you can say that this is often caused by a stress reaction, or perhaps this is often caused by the animal not eating as well because it's a high-dose group or something like that. The more you describe and the more you can know about it, the more you can feel that you're on safe grounds, either to proceed, or if you're not on safe grounds, to forget the compound. And then there's thirdly, there's a mechanistic thing, that if you know what's happening at a particular time of development of a particular cell type, you may know what the mechanism of this agent is acting in the testis. If that mechanism might be different, for example, if you have a one-species effect, you know, most of you have seen this. One of my species has effect, the other two don't, or the other one doesn't have an effect. Maybe that species is doing something slightly different than the one species, and you can explain it, and you can then have a registration which is, it's not sticky. It doesn't have any unexplained ends to it. So I think the biggest reasons are liability, producing a drug that will get through regulatory agencies, because the protocols are not, or the risk assessment and the testing guidelines are not written this way. They're written for more thorough testing. And I think that's an answer. Whether it's an acceptable answer or not, I don't know. Okay, let's proceed on and talk about Some additional things that would involve all of you as pathologists or toxicologists, because what we've learned in the basics of the testis now we can apply to pathology. Training of the histopathologist, that's why you're here. Certainly we will be talking further in this talk about training of the histopathologist, because as I go around to various companies and talk to them, you'll find that, okay, on the 2 days that we have a training course perhaps, at the end of the second day, you can pick up a lot of lesions that you've never picked up before in your microscope slides or that would have slipped by you before. But then what is your learning curve? As a period of time goes by, you may not see the testis for 6 more months or see a defect in the testis for 6 months, and your learning curve begins to drop. How can you as a company deal with that? Perhaps you can have one person become specialized in the testis, peer reviews all slides, or there are a number of ways, different ways to handle this. As a practical matter, all of you will not become testisperts or testis experts or something of that matter. A lot of you will forget what we're talking about today, but all of you will have a greater sensitivity as to what might be occurring. Normally in our course, what we do is we provide slides that are of an unknown nature. We know what they are, but we're asking you to look at these. And some of these are very subtle lesions, and some are not so subtle lesions. And we often ask, what % of these would you have caught in the routine fixation protocol that you have in your company, and what % would you not caught? And usually it comes out to be you would have missed about 50% of the lesions that are there, compound-induced lesions that we have because of tissue preparation and because of the way that you were looking at the tissue in the past. Well, we will be dealing with tissue preparation right here in a very simple way. Normally my lecture on tissue preparation can be as long as an hour or so, and I decided to include very few slides on this, but to just say that there are 3 basic plans of tissue preparation. There is the H&E formalin. Perhaps write these down. The formalin fixation plus H&E staining. Now that would be one plan. There is a possibility of now doing Bouins and staying with PAS. And I would include all of these in the first possibility. I believe that virtually all regulatory agencies are preferring this route, Bouins PAS. If most of you have seen the shrinkage that occurs after formalin, the tubules are shrinking away to a large extent. You cannot see the acrosomes. With the PAS staining combined with the Bouins, you get less shrinkage, and you can see the acrosomes of the spermatids. So this is minimum for testis nowadays. I think Dr. Clegg will probably comment on this. My comment is based upon what I've heard out in the field, what I've heard from regulatory agencies, but I feel that it would be useful probably. Would you like to comment now or later, Eric?
Lonnie, I was not planning to talk about fixation and embedding per se. I think you're as familiar as I am with where I stand, so why don't you just go ahead and say it.
Don't put you on the spot. But what I have found is that my prediction that you will miss in the H&E formalin, you will miss 50% of all effects that are out there. Are you used to missing 50% of all effects in other tissues? Probably not. The second fixation routine, this would be a step up, is methacrylate embedding. There can be a whole variety of fixations associated with methacrylate embedding, and methacrylate embedding with PAS staining. The advantage of methacrylate is that you get a much clearer resolution of the cell types present. There's no question about what the cell types are, and that you can do staging because the PAS technique will stain the acrosome of the spermatid. You can get an entire cross-section of the tubule on your slide. This is immersion fixation. You can go to perfusion and even do better with methacrylate, but I don't want to introduce too many variations here. And thirdly, there is perfusion with glutaraldehyde, epoxy embedding, and 1 micrometer sectioning after a standard staining with toluidine blue or methylene blue. Here in methacrylate, I would predict that you will pick up at least 25% more information than you had with protocol 1. And here you're almost picking up the remaining 25% here. The drawback with protocol 3, a lot of you are aware of the time, the expense. You're using more toxic compounds such as osmium. You only look at about 50 tubules a time. There are lots of drawbacks. So when you get someone who's been out in basic science and says, hey, you're not sensitive to industry, you don't know what it costs, how many animals we do, and all these kinds of things, I am sensitive to industry. And if I had to go back and choose a protocol in a routine testing of a compound of which I expected no reproductive effects, even in the high dose group, I would be right back here with the Boone's PAS. That's where I would be. However, one second, if I then suspected a reproductive problem, and this was the point in the evaluation where I wanted to do a real good job, I would do most of my animals with PAS, and I'd add approximately 25% of the animals and perfuse them in addition to that, and really check things out. That's the way I would do it. And I realize this is a discussion that can go on forever, because if we have 10 different companies represented here, we will have 10 different ways that it's presently being done, and 10 different rationales for this kind of decision. I heard one individual who told me that somewhere in the life of a compound, it should be investigated, the testis should be investigated well by histopathology, somewhere in the life of the compound. Whether you do it first or later, I don't know. It's a choice that's much more difficult. But somewhere in the life of the compound, you should be sure that it doesn't have a testis effect. There's a question, excuse me.
Just curious, with the Bowen solution, can you use a glutaraldehyde solution in the protocol there as well, and then use PAS afterwards?
Glutaraldehyde works terrible with paraffin embedding. It works terrible. And if you thought paraffin embedding was for formalin H&E is bad, then you do glutaraldehyde H&E paraffin, and it's really bad.
Leave our testis and glutaraldehyde for a day or two, and then we take them out and put them in formalin for a few days.
I'm not sure I've seen that, but the proof is in the pudding, and it looks pretty good. Okay, great. Oh, of that particular protocol? The thing is that I get confused because there's probably 30 different permutations of that, and that was the, yeah. If I, yeah. Okay, but I would like to look at it later. Good. Yeah. When you do this fixation stuff, the proof is in the pudding. What can you see? Then you stain it with what in the long run?
H&E and then PAS.
Mm-hmm. Mm-hmm.
Could you comment on the use of Davidson's fixation and compare it to the use of Boone's?
What is Davidson's?
I'm sort of in a fog myself. I know that it's used in place of Boone's in some instances. I don't know the ingredients. I just know that it's easier to use than Boone's as far as the rinsing procedures and
I'm going
less
to plead
toxic.
ignorance, but maybe someone in the audience can comment on it. Alcohol like formalin is what I'm told. Yeah.
So you're not familiar with
No,
how
I'm
it
not.
compares to?
No.
Okay.
Sure, sure. Please step up to the microphone.
We tried it at the time at Searle, and it did not give us any advantage over formalin. Although we were told that it is the thing to do, and we moved back to doing the Bowens, and that's my comment about the Davidson. We did not find any advantage. In fact, it was a disadvantage because for the testis specifically, we had to use a different fixative, more work, more protocol details and variations, which caused a lot of documentation problems. That's when we're increasing our possibilities of misdocumenting things. It did not happen, but nonetheless, we did not find any advantage to the Davidson.
I'm just curious, since we were missing half the testicular effects for a quarter of a century using formalin H&E, are there a lot of drugs out on the market, for instance, that cause infertility problems in men?
The comment there is made to, I think, to some degree to refute what I was saying. And I will say this, that if you have a high dose effect that you're missing, does it cause problems out there in the human field? The answer is I don't know, and probably you don't know either. But I also have an opinion on high doses, that high doses are there for a reason. They're there to provoke an effect. And if you can understand that effect, you can understand the potential for the compound, even though that may not be realized in everyday use. And I don't think a lot of us know the answer of what compounds out there that are unsafe. I have a personal belief that most of the compounds there are very, very safe that are out there. But that's only a personal belief. And I believe that we also are dealing with risk-benefit ratios and so forth. And for example, in our chemotherapeutic compounds that are out there, we destroy spermatogenesis every time. And we know about it. But if the information is there to be gained from a high dose, even though it is not applicable to perhaps human exposure or whatever, I think it should be obtained. The regulatory agency as well should be cognizant of what high dose means, I think.
If we're considering impotence as part of the whole picture of infertility, the story of the antihypertensive is fairly known right now in the medical community, and I just wanted to add that as a comment.
Okay, should we move on? I think, are the slides on? I think in terms of what we've said about fixation is that I've given you my opinions about testis fixation. It is an individual thing for your companies to experiment with and to play with, to get something that you feel comfortable with. There are no absolute answers to this. There is a menu of items that can be done. The power's off? Okay, thank you very much. I'm told I turned the power off. Okay. There is a menu of things that you can choose from to do with what you wish. Certainly, I'm not the regulatory agency. I'm sensitive to your costs, your time, how many tissues you have to evaluate as pathologists. I'm also sensitive to the regulatory agency having the information in an adequate way so they can make a judgment, and hopefully they can make an informed judgment. I'll just say that this tissue has been fixed with the best method I know how to fix tissue. It has pathology, a very subtle pathology in it that would have never been caught otherwise. And that pathology is a rarefaction of the nucleus in the spermatid. The question was asked earlier about a 2-week exposure if we pick up anything, and it turns out to be that Some compounds, many compounds, have a 2 to 3 week delay before there's any effect, before the effect begins. In most cases, we're safe because exposures are longer than 2 weeks. So, as a partial answer of that question, this has a very subtle defect that you see many times on many of your fixation protocols that we know that in the control sections this would not be present. I want to refer to some slides that Dr. Hess has and Dr. Chapin has. Each of these individuals have treatments in which you can put the slide of the H&E fixed tissue up and see no problem. You can then put the slide of the methacrylate up and begin to see the problem. You can put the slide of the epoxy up and really see the problem. And I don't have those slides with me. I wish I did, to really illustrate and bring home this point. But they are 2 different compounds that each of them investigated and found that the increase in the fixation protocol has allowed you to be more sensitive to treatment effects. Yes?
What was this on transmission? Did you put electron microscopy?
Yeah, we've looked at EM. The only thing we've seen is that these cells 2 weeks down the road will die. The current trend in tox exposure is shorter and shorter exposure protocols, which has implications upon where the lesion will be. If it was a testicular lesion and you had a long chronic exposure, in theory you could do spermatid counts of the epididymis and pick up a decreased sperm count in the epididymis. And I'm being told that this is not a very sensitive measure. You could do spermatid counts in the testis by homogenizing the testis and counting the spermatids and pick this up. The shorter the protocol regimen that's the ICH guidelines and so forth projects, then the more emphasis should be placed on the testis in determining where the effect is. We have to look at the timing of spermatogenesis, how long it takes, and what cell types we might be looking at. For example, in these fixation protocols I showed you here, degenerating cells are extremely difficult to see. They are slightly increased eosinophilia. You would not see them probably at low magnification. Here, they're slightly changed in color as well. But they are black as can be right here with this one. You can pick them up. We'll talk a little bit more about degenerating cells in a minute. The qualitative evaluation of the testis. I do not suggest pathologists change their normal routine except in a couple of ways. My bottom line conclusions will be that you probably need to go to high dry and examine a few things that you haven't examined before. And you probably need to do a couple of counts just for your own self to see that you're not getting a phenomenon known as failure of sperm release. Otherwise, your examination, your traditional examination at low mag, going across the testis, scanning back and forth, is a great way to begin. Just to add a little bit more to your examination than you have in the past. Don't forget the area between the tubules. We've stressed the tubules so far. This is a mononuclear infiltrate between the tubules. I show these slides here because these deal with Leydig cell proliferation events, a very hot topic right now because of perhaps the relevance of Leydig cell problems in experimental rodents to Leydig cell problems in man. No one knows this relevance. There's not enough research out to determine if, for example, if you see Leydig cell adenomas or hypoplasia, whether that is relevant to the human. There was a conference not too long ago that took place at Eric. Would you like to tell us a little more about that conference, or you just want to?
Actually, a significant part of my presentation.
Okay.
Will be the outcome of that conference.
Which dealt with the Leydig cell problems and its relationship to man. Certainly, all of you are aware of Fisher rats and the problems as they age and Leydig cell problems. Look at going back to the seminiferous tubule. This is sloughing that can take place. It can take place in a very similar manner if the testes are actually are taken out with the hands and sort of rolled around a little bit and before they're plumped into fixative. You have to be very careful in the way the rat testis especially is taken out and put into fixative, or you will get this kind of sloughing phenomena. Then you're left with the possibility of having to compare sloughing in your controls, hoping they were handled as roughly as your experimentals to see if the sloughing occurs. It is not hard to get sloughing in the testis. It's not hard to get sloughing in the testis if you cut the tunic when you fix the animals as well. Some of you may be cutting the tunica to let the fixative go into the tunic. Sloughing is a very common problem in this kind of situation. This is chemical-induced sloughing that is actually occurring, and we see slough cells actually going into the lumen. And electron microscopy, we can see a view of sloughing actually taking place. The most sensitive indicator of real sloughing versus trauma-induced sloughing is going to the epididymis. If you go to the epididymis and you see debris in the epididymis, then you're going to have the high probability that that sloughing was due to the treatment effect. In fact, I have seen situations where testis looked entirely normal to me by histopathology examination. Going to the epididymis showed that sloughing was occurring, and very specific sloughing was occurring. And then I went back to the testis, and I could find evidence of where the sloughing was occurring. So your epididymis is more useful to describe what's going on in the testis than it is with epididymal pathology.
In that case, though, how do you know that it wasn't an earlier injury and those are residual cells?
You're going to need to rephrase that again.
So you have cellular debris in the epididymis.
Yes.
You talked about how long the sperm stay in the epididymis, and you said that in the head and body, it may be a long time. So maybe those degenerated cells are residual in the epididymis and do not have current damage.
Okay, in other words, the damage is just passing through the epididymis, and the testis is no longer damaged, it's passing through. But that's useful information in itself that you did have damage. If you're doing a continuous treatment protocol, you should pick that up on a continuous basis. Okay, I'd like you to look at this slide for just a second. What's wrong? Just tell you right away, this is very high magnification, light microscope, epoxy embedded material, not what you're used to looking at. What's wrong? Vacuolation, no. That's normal. You're at such a high magnification, this vacuolation is normal in elongate spermatids. You've missed the obvious thing. The obvious thing is we have 1 or 2 cell layers missing right in this region. Okay, how would you have known the cell layers are missing? Because if you had understood staging, you would have known the cell association that should have been present and known that there have been giant Pachytene spermatocytes or perhaps spermatids in this position. How could you have pursued this? Well, if this is a common effect in all tubules, you can go back to see where the last viable cells were present. Obviously, they've degenerated at some point in this epithelium, and you've lost a whole missing layer of cell layers between very early spermatocytes and very late elongate spermatids. Again, the point being, knowing cell associations helps you know and predict what's going on. Okay. This is something a lot of you have seen, multinucleation of spermatids. The cell almost always multinucleated is the spermatid population of cells. This is caused by the opening of intercellular bridges that we saw earlier, and one doesn't always know the original cause of this. The original cause of this may be a fact that the cell is not getting its nutrients from the Sertoli cell. Its lowest energy state is not to maintain those bridges, but to open up. It may, on the other hand, actually be a germ cell defect. We can't always trace to where the origin is. I have seen these described as just multinucleate giant cells in the testis, or foreign body giant cells in the testis, which I think nowadays we wouldn't make that mistake. But here are very typical spermatids in a giant syncytial body. All of these came from the same clone. These probably are in a separate clone. This is a very unusual situation here with spermatocytes in a giant syncytial clone here. Very common disorder. This is a Sertoli cell only tubule in a testis. This is the situation I referred to earlier where only Sertoli cells are present. A very common situation where you have a complete loss of germ cells. Sometimes you will occasionally see a Spermatogonia here around the tubule in some of these. They may or may not go toward recovery. This is an unpredictable thing right now, what the recovery potential of this is. Here's something you see a lot of the time. Everything looks normal except there's that one tubule that you now call atrophy. Well, it's a Sertoli cell only tubule, and often you will see one like it down here and around the tubule. And what that means is just one tubule that is Sertoli cell only, but it's been cut in several places as it bends back and forth. This is common on a recovery study where you hope that every tubule comes back 60 or 90 days later after you've discontinued treatment. But most came back and this one didn't. Why didn't this one come back? No one is quite so sure, but it's a very common phenomena to see. Okay, we have some pathology in this slide, a fairly subtle effect. Is this pathology here? Actually, this turns out not to be the pathology. This is a degenerating cell that is normally a degenerating cell within the epithelium. In meiosis, a lot of the cells do degenerate in a normal testis. The pathology here is the head shape of the spermatids, a very subtle defect. This illustrates 5 compounds that we gave in our lab to look at. We were interested in the effects of the 5 compounds, but this is a staging map. The staging map shows the cells that were observed to have problems here, and the cells that we predicted were affected by the compound. In other words, since we sacrificed later, we did what's called a backtracking to determine which cells were affected. The point, however, that I'm illustrating is this is a good way to communicate your findings if you need to go into detail about an effect. This is a way that everybody can understand this effect. You're either left with describing findings in a way like this or saying tubular atrophy or tubular degeneration. And I think in the future that won't be acceptable, as we explained earlier. Okay, here's some pathology of the testis. There are 2 or 3 things wrong on this particular slide. I want to illustrate, however, that this is the way degenerating cells appear in this 3rd protocol, the epoxy protocol. They appear very dark. They are hard to miss. They're unavoidable. And this was an RNA synthesis inhibitor that was used here. This was procarbazine, and in this case, the nucleolus of the Sertoli cell appeared abnormal also. And we had, even though this was given on a long basis, we had just a few pachytene cells. These were pachytene cells. You can see there's a whole cell layer missing between the base of the tubule and the spermatids, and that's the pachytene cells that are missing. Recognition and quantification of degenerating cells. There is a concept that I'd like to produce that's called go or die. A spermatogonia, once it has its very first division, can do only 2 things. It can go all the way to form a sperm, or it can die. It doesn't have any other choice. If it dies, it's a nice signpost for you within the epithelium to determine what's going wrong. And our lab has been especially involved in a research capacity to look at degeneration of cells in the epithelium. The cells that normally degenerate within the epithelium are those with crosshatches on them. A lot of spermatogonia degenerate very early on. A lot of spermatocytes degenerate during meiosis. This is the slide I showed you earlier of the meiotic arrested cell, which I said was normal. There are a few of these cells degenerate, but a very, very, very low numbers of these cells are degenerating. But you have to have the normal pattern as a background. Seeing spermatogonia degenerating has a big impact on spermatogenesis. You're really looking at what in your tissue is above the background level. These are degenerating spermatogonia. They degenerate by apoptosis, very classical features of apoptosis. Anything beyond spermatogonia, although they tunnel end label, which means they have the characteristics of apoptosis, doesn't look like degeneration apoptosis. Spermatids, spermatocytes, although they biochemically have apoptosis, have features that look like necrosis in the testis. It's not generally known, but only spermatogonia, the two end cells here, have those typical apoptosis features. Here is a spermatid. They call this apoptosis, this method of degeneration here. It doesn't look like anything like it, but it does end label, a tunnel end label indicating apoptosis. We can even identify this by light microscopy by the presence of a light acrosome that's present in the cell. And we can determine where during the whole pattern of spermatogenesis it degenerates by putting the degeneration on a staging map. Here's a meiotic degenerating cell which has a lot of speckly features, the chromosomes. This is apoptosis too. Apoptosis, as far as I know, has never been described in a metaphase cell, but that is. And here are spermatids degenerating out here, and their position and size indicates that they are indeed spermatids degenerating. The most common form of degeneration you see in the testis is the degeneration that occurs after sperm release, when sperm are not released normally. Sperm are normally released in stage 8, and they normally are right down in here, but after stage 8, in this stage, early stage 9 tubule, if there is a problem, and this is a fairly thick epithelium, you might, on H&E staining, say that's entirely normal. But by looking at it with better fixation techniques, you can see that it is stage 8. The sperm should be gone at this point. They have been taken up by the Sertoli cell and are being phagocytosed at this point. Only by knowing staging are you going to be able to detect failure of sperm release. Yes?
Lonnie, would you comment on the residual body, phagocytosis, inherent at the base of the Sertoli cell that looks like an abnormal degenerating cell?
Okay, well actually it's shown a little bit in this slide. When sperm are released, they have cytoplasm, the residual body, that's here, here, and here. And it looks a lot like degenerating cell. It's only present during a short period of time. The only way you can tell when that time is, again, by knowing staging. And it is present right at the time of sperm release and has been recognized by many people as degenerating cells when it is actually a normal part of the epithelium. This terrible slide shows vacuoles in Sertoli cells. I've got to get another slide, but an extremely common feature. These vacuoles, and most of the vacuoles you'll see in the epithelium, are near the base, and they are generally extracellular. If we really look closely, these vacuoles, here is a cell membrane here, are between cells in this area, this area, this area. Here's the cell membrane here. If you remember the Sertoli cell barrier, the Sertoli cell can secrete basally and luminally, but when toxic effects occur, it often secretes laterally between its junctions and opens up these giant round vacuoles. And when you see big vacuoles in the epithelium that are rounded, that usually means extracellular vacuoles, and it is a clue that the Sertoli cell has been disrupted. Quantitative evaluation of the testis. Most of you have probably never undertaken quantitative evaluation of the testis in any great detail. In talking to regulatory agencies, they like quantitation because your subjective impressions are often plus, 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 plus. Is his plus, 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 plus the same as his plus, 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 plus? And how many pluses could be had until it's, you know, this kind, I mean, In other words, if we have a quantitation of germ cell abnormalities, risk assessors can work with it. Subjective information is much less able to be worked with. Now, quantitation in the testis can be easy or it can be very, very, very difficult. For example, Leydig cell hyperplasia. I've seen Leydig cell hyperplasia called Leydig cell hyperplasia when it was not. When the tubules are depleted of germ cells and they're very small, you see lots of Leydig cells, but in a very small testis. You probably had the same number of Leydig cells you originally had. Now they're more apparent because the tubules, which occupied 86% of the testis, now only occupy 30% of the testis. So to determine actually how many Leydig cells are present is not an easy job. There's quite a bit of work involved there. But on the other hand, for example, to determine what failure of sperm release is, you could probably determine that on the microscope in a half an hour's period of time to see if your controls are the same level as your treatment group. A very good test to show a normality of the epithelium, because this is the most common effect you'll have in the testis is failure of sperm release. In over 50% of the defects I've seen that are subtle, where all the germ cell layers are present, failure of sperm release is present. And it can be quantified fairly easily. Let's look at some methods to quantify that have been used. Eric, do you happen to know what species this is right here that this was done on? Is this his domestic animal paper, or I think I've got the latest rat data, but we're looking at testis weight here and how many replications are needed, how many animals are needed to give you a 90% probability effect. And if you're looking at, let's say, a 150-day-old animal, we don't work at the 90% probability. We work generally at the 95% probability level. 30 animals are needed to give you a 90% probability in this situation. That's a lot of animals that are needed just for testis weight to determine a difference. Here we've counted spermatids per tubule. And we've asked the same question. And this is rats, this is rabbits. Let's focus on rats here. And we need 30 animals. And here at the 90, little less, 20, close to 30 animals at the 95% probability level to pick up a 10% difference. Very insensitive measures. Many people advocate, I don't know why this slide is, I'm going to see if I can go back and then back to it and see if it's, for some reason it's slipping down. But many people advocate stereology. Once your company enters into full-blown stereology, you're into it for a good 2 years. And you probably will never emerge from it healthy because it is extremely laborious technique. For some projects, you may have to enter into it. You take a specimen, you use light or electron microscope techniques to give you pictures in which you put test systems over. Now, a test system, looks something like this. Lines or curves or points. You put testis systems over your micrographs to give you counts. You plug these counts into formulas to get results which are amenable to statistical analysis and give you conclusions down the long run. And stereology will tell you, for example, how many Leydig cells there are in a testis, or whether you really do have a Leydig cell effect. It will tell you how many spermatids are present. But it is extremely long and laborious technique. Damage detection and mechanistic interpretations. Most of you are used to doing damage detection, but you're not used to doing mechanistic interpretations. Morphological pattern of response is a concept that I developed about 10 years ago. And it was based upon some test compounds that we put through and tested animals on. And I think it will become very valuable as time goes by in the toxicology world. Let me go ahead and explain that. Let's make the supposition, and it's not too hard to make the supposition, that biochemical events in a chain lead to a morphological outcome. Now that's not too hard to realize, that virtually all, we're talking about pathology, that's morphological. But what led to the formation of a spermatid, for example? A multitude of biochemical events in a chain-like fashion. Not necessarily in one chain, but there may have been several chains carried on simultaneously, such that first event goes to second event, goes to third event to get normal spermatogenesis. If you take an agent and you interrupt first event, you have often abnormal B, and if it doesn't correct itself, it has abnormal C, and it leads to some kind of response in the testis that I called a morphological pattern of response. Now, what does that mean? It's a cluster of events you see in the testis. For example, one of the most common morphological patterns of response are disorganization of the epithelium, sperm tails going every which direction, vacuoles, and failed sperm release. That's a very common cluster of events that occurs in about 50% of all lesions. And that's what I call the morphological pattern of response. Well, let's take another agent. Suppose agent X is well known. It's something that we know about. It's used in laboratories as a specific inhibitor of this process. Agent Y might be your drug that you're testing. It does the same thing, and it gives a shared pattern, morphological pattern of response. Isn't it not reasonable as a tentative hypothesis to believe that this agent may be acting in the same pathway as this agent? And we can then begin to know about mechanisms in this manner. We can interrupt the second event and get the same response. Now this has been tested. Well, I'll just go through it and look at one of the early tests of this particular thing. We looked at colchicine and vinblastine on the seminiferous tubules of the epithelium. Very specific inhibitors of which there is lots of cell biology information about how they affect microtubules. Then people have come along and looked at, for example, Benamil. Benamil is a herbicide, and it has some microtubule effects. And it also shows the same pattern of response in the testis as vinblastine and colchicine, a common shared pattern of response. Not only does that occur with this agent, but I've done experiments with hypophysectomy. Take out the pituitary. Well, it's very possible that many of the agents that you use to test drugs will decrease pituitary levels of LH or FSH or affect the hormonal system in some place. So this would be a good model. So when you take out the pituitary gland, you get a morphological pattern of response in the testis, a very specific one that you can place on a staging map and that you can identify as very characteristic. Well, what if you use some other agent? You don't take away the pituitary, you take away the binding of testosterone to its receptor. Use cyproterone acetate. Do you get the same morphological response? The answer is yes. What happens if you knock out Leydig cells with EDS? You get the same morphological pattern of response. So now there are about 10 agents, and recently we looked at streptozoitis in diabetics. We have a slightly diabetic rats who have a slightly decreased level of testosterone, and very subtly you can see the hormone effect in the testis. So that's a valuable, if we get enough of these morphological patterns of response out in the field, then it's a valuable asset to you to be able to say, hey, I see this, we better go back and measure hormones or see what's happening in the hormonal route of the testis to be able to understand what's going on. There is a gentleman by the name of Paul Cook at the University of Illinois who was playing around with giving animals, making them hypothyroid right after birth. And when he did that, he got a testis in the rat that eventually became twice its normal size. Well, that's a pathology as well. I guess a lot of men would like to have the testis normal, twice the normal size, or whatever it might be. But nonetheless, we also found a recent compound just published the other day that did the same thing by making the animal hypothyroid. You have a model compound which you know the specific effect, then you have another compound which you're testing. And if you can compare the two in the literature, it really makes it nice to see the morphological patterns of response. Window of effect. If we have a dosing protocol, let's say, that's 2 weeks long, and no sacrifice, and let's say we on our staging map out this effect as occurring here in the late spermatocytes and the early spermatids. The effect is as long as the dosing protocol. It meant that it affected only this cell type here, and during the dosing protocol, that was transferred to a wide band. Well, what happens now if we let, we've talked about this already, 2 weeks go by as an interval after dosing? That effect has now moved down the epithelium by a 2-week period of time. If we let 4 weeks go by, it is now in the epididymis, is displayed by fewer germ cells in the epididymis. And now, 6 weeks, the effect is out of the testis. So in designing protocols, one has to think about the sacrifice interval and where the effect is going to be. The short-term protocol studies that are being proposed and used now by FDA and so forth started out as a proposal from the Japanese to have only 2 weeks of exposure of the animal and then 2 weeks before sacrifice. That's the way they started out. They're not that way now. This is now modified by FDA to increase the exposure interval in the ICH guidelines. But if you have 2 weeks of exposure, let's say to Spermatogonia, which are around for 16 days, could you as a histopathologist pick up an effect on Spermatogonia after 4 weeks when that would be displayed in the spermatocyte population of cells? Just something you're not used to doing, to go in and focus in on the spermatocytes. So the shorter the term protocol, the more important it is to have a keener eye in histopathology. The effect on the Spermatogonia and the spermatocytes, which most of you missed earlier when I showed the slide that you picked up vacuoles instead of the missing cell layers, would have wiped out the testis if you had wiped out those cells. In a longer period of time, the testis would have been completely gone. No germ cells present. Yes, sir. This is a little chart in our book, and it allows you to predict, or backtrack as I call it, which cells were affected by an agent that was given on a single administration. I realize that rarely do you ever give a single administration of an agent. Our lab has done that and on a number of occasions. But here is a predictive effect a table. Let's just say this is the weeks of necropsy, and that we're dealing with approximately 4 weeks after exposure of a single administration of an agent. Let's say you find at this time at 4 weeks the step 7 spermatids are gone. You come up to 4 weeks and then step 7, which would be right about there, draw a line parallel to that, and it would indicate or suggest that the intermediate spermatogonia were the sensitive cells at that time. This kind of a chart is not very valuable for most of your testing protocols. And backtracking is not very valuable because most of your protocols are based upon a much more chronic administration of an agent. But we are all heading towards shorter and shorter and shorter administration of an agent. I don't know if Dr. Clegg will tell you, but he told me last night that EPA is recognizing that most exposed individuals are not exposed on a chronic basis. they're exposed periodically. And I may be talking off the top of my head and may be wrong, Eric, and you can correct me later. But this is one rationale in EPA maybe for doing a shorter term exposure, is because to mimic human exposure. This is certainly different in FDA. Here are some questions that can be asked about histopathology. And a lot of these require different designs of experiments. And I'm sure there are the standard designed experiments, and then there are the special experiments that you do for one reason or another. Hopefully not because the regulatory agent asked you to go back and do them, but hopefully you did them initially. When are the first morphological changes evident in the testis? To do this kind of an experiment would require short-term experiments and serial sacrifice. But it is extremely valuable from the standpoint that if you're knocking out the testis, if you find the first things that occur, you know where the vulnerable sites are. Where does the interruption of spermatogenesis occur? It goes along with number 1. It can only be done by using staging. What is the morphological pattern of response? Only be done by using staging. What is the likely mechanism of an action of an agent? I described to you a minute ago this morphological pattern of response type of thing, where we can begin to get some handle as to the mechanism of action of an agent. Then we go on to something entirely different in number 5. This is what you do in most of the time. What is the maximal damage possible after the continuous or intermittent administration to an agent? We even go out of our way to do that. We give doses that are extremely high, in which many-fold greater than we would ever expect an agent to do, and we give it for a long period of time. This is your typical exposure. But you don't look at how the damage progresses, generally, which would require serial sacrifice, and what is the timing for complete recovery. If you do have an effect, of course, that's a very important thing that most of you do. Does the testis recover? How do you deal with 1 or 2 tubules that don't recover? Is recovery complete? All of these require different designs. Now, there are different available endpoints that you can use. And again, this is a menu. This is not a suggestion from Dr. Russell to use. This is a menu of items that people are currently experimenting with. If you go to one company, they say flow cytometry is the answer of the testis. You go to another company, they say, well, we homogenize the testis and count spermatids. You go to another company, they say, we don't do any of this. We're doing something else. We rely on some other feature. But this is the menu that people are selecting from. Some are very sophisticated. Well, behavioral studies we haven't talked about too much, but this is something that we've just alluded to, that you can homogenize the testis in a blender, put in a little detergent which disrupts the membranes, and find the spermatids whose nuclei are not disrupted by this treatment because they're linked by disulfide bridges. And you can count them, plug them into a formula, and determine daily sperm production in the testis. All that sounds good, except the standard error is very, very high in this kinds of experiments. Coefficient of variation being somewhere around 15 to 25% on this kind of situation, and difficult to make conclusions from. But this is one of the more common procedures. This can also be done in the epididymis, and I'm also told it is much more accurate when you're looking at cauda epididymal sperm counts. When you homogenize the epididymis, the cauda of the epididymis, you get a much more accurate count about what's called sperm reserves in the epididymis. Flow cytometry. Flow cytometry has been used by a couple of companies. And what you basically are looking at is the nuclear content of the testis cells. So spermatogonia, which have a 2N nuclear content, are grouped together with Sertoli cells. And you have a 4N and then 2 types of N counts that you get, which are the spermatids. And those 2 types of N counts, the 4N and the 2N, are all put together in a bar, and you look at the relative proportion of those cells there. Of course, if all the cells are relatively lowered in number, you won't get any pattern change and no effect. But it's described as a quick screening effect to see if one or another of the cell populations is out of whack. Pretty expensive and sophisticated technique to do quantitation. Counts, morphology, we've talked about this, a very time-consuming technique. Sperm motility assessment. Many companies have described, are we or are we not going to buy the software, the microscope, the video camera to do on a routine basis motility parameters and this sort of thing. If you do enough motility parameters, and believe me, these machines can give you numbers of which you can't comprehend and understand. There are so many different parameters that you can get. I guarantee you will find a significant difference somewhere along the line just because you get so many different values. My suggestion is that you rely on some of the basic parameters if you have these machines, and I'm not an expert with this, and perhaps Eric can tell you much more about it, is to rely on some of the very basic parameters and don't get a lot of numbers from this machine that will cause a lot of confusion because the functional significance of these numbers is often not known. You can penetrate a hamster egg with the sperm from a lot of species. That determines whether the sperm is functionally good or not. Very expensive and time-consuming. There are in vitro methods that are ideal for mechanisms but don't lead to many practical applications in standard testing protocols. And of course, there are endocrine measurements that you can do if you suspect endocrine disruption. I spoke about this a second ago, homogenizing the epididymis, counting the homogenization-resistant spermatid heads that are present in the epididymis. This is often a very, very good way to do a testing protocol. In fact, since I do not know of any lesions in the testis that affect the left testis, preferably over the right testis, or the right over the left, you can design experiments where you take one testis for histopathology and the other testis for this kind of an experiment, and actually can do some correlation since you're using the same animal. So, what are some endpoints for future use? And I can guarantee you we're not going backward in histopathology, we're going forward, and that's always been the trend in the past, and probably we will not be reversing ourselves. Improved histopathology using staging and specialization of tasks. What I mean by specialization of tasks, I'm certainly aware that even if you do staging for a few days and you go on vacation, you've lost most of it by the time you come back from vacation. You need a reference guide. You need to refresh yourself periodically. But you may not even need to do that. In your company, you may have someone that is looking at the testis slides. You may have someone else who is peer reviewing them that are specialists in this, whom you can refer to. If you have 6, 8, 10 pathologists, you may be able to afford this luxury. But at least all pathologists can have their eyes open a little bit to testis defects as a result of some of the things that I'm saying. You can be a little more sensitive to those effects. Oops, didn't mean to do that. Automation of testis cell counts. Where we need to go is we need to be able to put a slide of the testis under the microscope and let a computer count cells and tell you that that treatment effect the treatment testis has the same number of cells as the non-treated testis. That would be nice. When you submit this to a regulatory agency, there's little question that spermatogenesis is absolutely complete if you could do automated testis cell counts. Our lab, we've proposed to do this kind of thing in the past. We've never received the money to do it, but we have an excellent proposal to automate testis cell counts, at least semi-automate testis cell counts. Computer recognition of degenerating cells, fertility studies we need fertility studies that are much more sensitive than we've ever had in the past. There's been a lot of work at NIEHS on fertility studies, and it really takes a compound that knocks out the testis almost completely before you have any fertility decrement. Targeted endpoints that we need to improve those, and we need combination studies where we take an animal and get better use out of it than just histopathology all by itself, perhaps sperm counts from the same animal, and other information, perhaps endocrine determinations from the same animal. We spoke about task specialization and how you might accomplish that in your company. I'm not sure what the best way to do that is. I do know that when I've been to companies and we compared before I came and after I came, there is always the realization there is a lot more to know. There is always the question, like was brought up earlier, well, is that really going to help us make a safer compound in the long run? That's a valid question. Usually, after a year or two later, we don't quite look at the testis the same way we did before. We're looking at it in a little bit different way than we did before. And some people will say we have taken one or two people and devoted to the task of looking at the testis. There is something that I told somebody in the break today. All of you got a break, but I didn't get a break today. I just got a chance to quickly drink a glass of juice. But talking to people about the testis. Okay, I lost my own point there. It's gone. Excuse me. It'll probably come back. I made up several slides that I think are good questions. Unfortunately, I don't have good answers to all of them. But I think these questions are worth discussing for the future. I have some opinions about some of them, and I'm sure you have some opinions about some of these. But I've got about 10 or 15 slides of the most common questions asked me when I go out to discuss evaluation of the testis. This question I've sort of answered already because we hit this point earlier. What % of lesions are we missing in the testis? Perhaps the second question should be, what is the meaning of this? And clearly I know less about the second question than I do the first question. But I do know we're missing testis lesions. How it's interpreted by the people in your company, especially higher up people, or how it's interpreted by the regulatory agency, it's my opinion that regulatory agency people ought to be at these kinds of meetings as well. Because if we look at both FDA and EPA, there are very few people who can interpret a lot of the data that you put forward. It's usually the sticky parts of your application which cause problems, and I use the word sticky parts. If your registration comes through with some open gaps, which you can't explain by the literature, or which you haven't looked at very closely, that's one of the most common causes for either kicking it back or for failure of the compound to go through. If you have spent how many millions of dollars to get the compound to the registration point, and you have 2 or 3 organs that are sticky in the high dose group level that you really want to explain, again, this may seem self-serving, but you need to get somebody in who knows the literature and who can put this in the right perspective for your company. And it may not turn out that there is a right perspective for your company, but I do believe that there are. I was called a few weeks ago by the FDA, and I was read a company's, without knowing which company it is, I was read a company's of testicular effects. And I was asked, do I know what that means? Because the person said they are not a testis expert. And I couldn't read it myself. I couldn't begin to tell you what they had found in the testis because it was not written well. I don't know the fate of what happened to that registration certificate registration, but it's certainly my answer didn't help them one bit. But make sure that there are no sticky points left in the registration. Now, whether you as a pathologist, at least you as a pathologist can, from your level, make this as good as possible. What happens at a higher level, I don't know. It's up to those people at a higher level. It's their responsibility. But you can give your report as good as you can. Is the effect on the testis direct or indirect? Okay, Dr. Russell, we have a compound which affects our testis, and we would like to know whether it's direct or indirect effect. It's a hard question to answer. One of the ways you can determine a direct effect on the testis is inject the substance directly into the testis. You can give that compound at 1/100th the normal dose to, now this will not work with many pharmaceuticals, I realize that, but we have had people do this. In a non-metabolized compound will affect the testis directly if you inject it directly into a ras testis, and it can be moved throughout the testis very quickly and very uniformly, and you can determine it. Compound at 1/100th the normal dose would probably not go to the liver and metabolite come back and affect the testis. There is a lot of unknowns about this, and a lot of companies want to know because they will have an effect, and they are hoping it is an indirect effect rather than a direct effect, and they want to discern the two, and it's not always easy to tell. Who should peer review our slides? Should we designate one person, or should it be just random peer review within your company? Or should we send them to an expert to do that? It's a question that has to be individually answered. But I recommend if you have 4 or more pathologists in your group that one person really become more of a testis expert than the other people and do see all of the slides that come through on the testis to peer review them. It can be handled in many different ways. When we use dogs for histopathology, we often get immature animals. Is there something we can do about this problem? Yes, get mature animals. Well, I realize it's not quite that simple because I've even seen regulatory requirements which specify dogs of particular ages that are immature. So what you're dealing with then is describing the pubertal spermatogenesis in a dog and the effects of a compound on it. So both the regulatory agencies and the people who supply the animals and the people who order the animals should be aware of this. You always hope your immature dogs are equally distributed in both groups and that you're really sure that you can tell what's maturity and which is a pathological effect. I'm not sure that's so easy. And there are some problems if you ever use rabbits with stress reactions in rabbits and histopathology as well. But certainly this is a common question. What does the regulatory agency mean by staging of spermatogenesis? I think I've covered that, but not as much as I want to hammer it in. It means that if you know staging, you can perform your normal pathological exam and maybe go to high dry after you do that and take a few looks around for missing cell layers or failure of sperm release. And you can do that only after you know which tubules to look at, and you can only do that after you know staging. So either you're into the atrophy description or you're into the latter, a more informed description of spermatogenesis. You can go either way. I think the atrophy will cause problems in the long run with the regulatory agency. It does not mean that you go to your compound-treated animals and do the frequency of stages. That's not what's meant by the regulatory agency. And many companies had been worried, especially FDA-regulated companies, by the way that that guideline was worded. It was very poorly worded. What are some subtle testicular lesions that may be signposts for testis effects? Well, if you're doing Booins and up in terms of fixation, you can certainly look for failure of sperm release. One little problem is sperm release is not even perfect in the normal animal. And the way I quantify this is I quantify the sperm that are not released per 100 Sertoli cells. If you count 5 tubules, you're going to get about 100 Sertoli cells. And in the normal animal, about 2 sperm are failed to be released in the normal animal. And if that goes up, double, triple, quadruple, you have failure of sperm release. That's a very quick down and dirty assay. When should I get a consultant to read the slides and interpret the data? In my case, well, my answer is every time. I think the real answer is, if you've invested in a compound and you want to make sure your registration, the wording, the pathology, that there are no sticky points to it, and you're dealing with a situation where the benefit, where your risk-benefit ratio is such that you should have a good testis in the particular compound you're looking at, then you really need to make sure that your pathology was interpreted correctly and that the best light was put on it. I've seen many cases in a high-dose group. The animals are not eating, the animals are undergone stress for one reason or another, and you get failure of sperm release in your controls like mad, and you get it in your experimentals. The regulatory agency wants to say, well, was it increased in the experimentals over the controls? Or maybe it was only noticed in the experimentals, where it occurred almost the same way as in the control. Maybe there was other things. What do vacuoles mean? What does a few multinucleate cells mean? You know, how common are these effects? What cell are they likely associated with? That sort of thing. What if I have background? Well, this was. If I have background effects, how do I handle this? And this is extremely common. We did have some animal suppliers in the past that were supplying animals that were Sertoli cell only in about 1 out of 6 out of 10 animals. They were Sertoli cell only, and the companies were using this, and they had a background problem. I've seen background problems in inhalation experiments where the animals were restrained or at temperatures in which they were uncomfortable. I've seen background effects in animals which didn't eat. And of course, when you're in a high-dose situation and you're giving something orally or something of this nature, that's a very common situation that you have to really deal with very carefully. I have a testicular effect in one of the 2 species in which the compound was tested. What does it mean? I've seen this extremely frequently. I don't think I can answer this question, except that if you can show that the species where you have the testicular effect is less related to man by the kind of effect. And you can't describe that by atrophy. You have to be very specific to the kind of effect. For example, steroidogenesis may be different in one species than another. And the species of where spermatogenesis was good was the species you want to compare to human, and you want to rationalize that. I think you really have to know something about it. What is the most common problem associated with registration of compounds from the point of view of the testis evaluation? Well, that is the sticky effect that I referred to earlier. Unanswered questions, effects that are not explained, not literature justified, and not done with techniques such as good fixation, of which you can really tell what cells are damaged and where the effects are. My computer allows me to categorize testicular lesions, but you have given me many more names for lesions than are now available to me. How should I go about doing this? Well, I remember one company I was at, I had to sit down, didn't have to, I was pleased to sit down and write out different lesions than were available for the pathologist. I think people need to get together and publish a whole new set of lesions that we now know occur in the testis, or more specific descriptive features that where you can plug into your computer, as most of you do now in your company, and do routine histopathology. That certainly is needed out there. Never asked, but should be asked. What magnification should I be examining the testis? It's never asked because most of you put slides on. The older pathologists don't have even any XY movers on their slides, am I right? And the newer pathologists do, and you zip through, and you do a good job looking through at low mag. Most of you don't have oil immersion lenses. Now, I'm not saying use oil immersion. Don't get up in an uproar. But to go to high dry mag for the testis is almost a necessity. It's almost a necessity. So I consider the scanning that you normally do plus a brief look at high dry as very important. Risk assessment. Garbage in, garbage out is the old saying for risk assessment. The better the information you can give the regulatory agency, and I'm sure Eric will be speaking on this today, the better and a more adequate job of risk assessment that can be done. I think where companies and governmental agencies are in dispute is that both of them need to focus on one thing. How do we make sure the compound is not harmful? And if both of them did that, and even in joint meetings, I think we would come up with a much better understanding of each other. I do know that there are many poorly worded registrations going out, and there are also very poor interpretations of those at governmental agencies, and unreasonable requests many times that come back from the regulatory agency. There is no harmonization of these two groups, and it really does good to, I think, in the long run, to get not just company people in a meeting, but to get regulatory people together with company people so they can understand each other. Both will go back to their own domains and work toward a common goal. I think that's the answer. I recently gave a talk in Brazil on this topic, and I said, well, what kind of regulatory agency down here do you have in Brazil? And Brazil has a population the same size as our country here, and not a little less than our country, but a land mass the same size as the United States, and if it weren't for Alaska, they would be bigger. And, oh, they said, we rely on you. Well, you means Europe, Japan, and the United States. And there's so much of the world that actually does rely on us for that. It kind of impacted me how important the job that all of you are doing in getting chemicals out. So this is another sunset that tells me that my portion of the talk is finished, and I'll be happy to answer any questions. I'll go to a global answer to that and then go back to a specific answer. There are certain things that, for example, you don't call it the PMA anymore. You call it something else, don't you? What is it? The MRA. MRA, okay. They need to work out. They need to have somebody carefully look at what a stress reaction is and what the morphological pattern response is. I believe I know it, but I haven't seen enough common situations where we can say stress due to this, stress due to that, stress due to that. Do they all have a common morphological pattern response? I think you can get a lot of failure of sperm release and some vacuoles and Sertoli cells and a little bit of a disorganized epithelium. I've seen an inhalation study that was absolutely terrible because of probably physical restraint stress in an inhalation chamber, which mostly doesn't occur now. The animals are allowed to have free mode. It was really terrible in the control group. But that has not been done. And stress due to inanition has not been done. There is some literature on this, but a carefully worked out study has not been done in well-fixed tissue.
My other question was, in the Burnson paper, they knew that they're going to look at stage 7 tubules. And so you have to look at like 12 tubules from 8 animals, and that's when you know what stage you're looking at. For the people who do go down to like a high power, I assume you advocate looking at every type of tubule stage, at least how many tubules of each specific stage, so you kind of get an idea that you're going to see something.
Well, no, I don't think you should look at every stage. In fact, if you think about it, where are sperm released? If you're watching the sperm charts, sperm are released in stage 7 and 8. Okay, so anything that transpired to any of those cells earlier than that is going to show up in those stages. And so I suggest you focus on the 7 and 8 and then the 9, 10 right afterwards to see failure of sperm release, but the 7 and 8 to see the final product of spermatogenesis.
There's a statement that, and I know I've kind of felt that you can't have a unilateral lesion. I mean, and most compounds don't cause that. We denervation or vascular type things that you think you could see that? Do you have any information on seeing that?
That's kind of news to me. One company once told me that they thought they had a unilateral lesion, and when we looked at it, it was something like only 3 animals in which you could, out of the group, showed the effect, and it was basically on one side. But when we finally talked about it statistically, it was like having 3 girls in a family. I mean, 1 out of 8 probability or something like that. So, but no, I've never seen the effect being unilateral. Varicocele and so forth can occur sometimes on a unilateral basis. That is a vascular-based disorder, and you have slight differences in the vasculature on the left and the right. But basically, I haven't seen, never seen a clear, proven case of unilateral lesion.
Well, if I could just say something on that. I think when you came out to SIBO, I mentioned to you that I found spontaneous degeneration in the rat testis more commonly, much more commonly on the slides, actually.
That something sort of related to that, people say that peripheral tubules in the testis are always more affected than central tubules. Have many of you seen this? And there is no good explanation for that except in intraperitoneal injections regimens, which is not a typical protocol that you use. But if you give something intraperitoneally, it travels down the inguinal canal and may penetrate into the outer tubules. But that's not a good explanation for why most of those are on the sides of the testis. But if anybody has a clear unilateral testicular lesion, whether it be aging or whatever it is, I'd like to see it published and really be able to look at it closely. But of all the lesions all of you have looked at, it must be in this room a lot of combined testicular lesions you've seen. We're not seeing anybody said, yes, I know of one definitely. The testis, left testis absolutely wiped out. The right one is good. You know, we don't hear that at all. But usually we hear more subtle defects, and I'd really like to see a study done. But aging would not be a pharmacological effect, but what would cause that, I don't know.
Just to address that issue, I've seen studies where in recovery groups, for instance, you will have a clear-cut response and it will be bilateral, but in that same group you will have a much higher incidence of unilateral lesions than you will in control. It's always confusing as to where to place those, and that's the
Right, and that unilateral lesion may not be the right always. It may be the left some of the time.
It's
right, and that's what it's a little bit different situation. And I've seen that too, that one testis may recover almost fully and the other one just, you get the Sertoli cell only tubules to a large extent.
Do you have any handy hints for differentiating the pubescent testicle from compound effects, especially in primates? It seems to be a problem in every one of the 50% of the lesions that I pick up. You think it's that high, John? I know it'll vary, but...
Usually, pubertal animals have a very inefficient spermatogenesis, and you get degenerations along the way. And these are sporadic, here and there. But if you've got a lesion, usually you have an absolute barrier at some point to spermatogenesis. It's not a very good answer to the question, but you've defined the problem very nicely in monkeys, and that occurs in dogs as well. So.
We've had that experience too, of course, as we all have. And I think that in those instances, one has to consider the weights. They have to consider what the prostate looks like. They have to consider what the prostate weight is. They have to look for the presence of supposedly mature sperm in the epididymis. You have to consider the whole male reproductive tract when evaluating puberty. And with monkeys and dogs, you're wading off to deep water unless you do that each time.
Right. In other words, what you're saying is if you have sperm in the epididymis, that is a pubertal animal that has been disrupted by a compound effect. If you don't have sperm, it's a pre-pubertal animal.
Depends on maturity of the prostate epithelium.
Which is a testosterone effect, and that's growing with time.
Go ahead. Before we go to the problems of the prostate and puberty and maturation, go ahead with your question.
Just two questions regarding risk assessment for the so-called subtle effect that you talked about. Those can be many. What's your recommendation with regard to the impact of those on the risk assessment process? Would you suggest that companies do more at that point, even if it means additional studies to assess the impact, or should they just attempt to give it an explanation and go ahead and move forward?
There's a certain phenomenon. Should we find out information or should we bury our head in the sand? And my question is that we should find out information, but learn to use it wisely. Usually, the subtle lesions are the high-dose lesions, and usually you're getting spermatogenesis decreased, 20, 25%, 30%, and regulatory agencies as well as companies should know that to be able to put this in perspective. If it doesn't occur at your middle or low doses and so forth,